Hello friends, I am Abhishek welcoming you all once again in the channel Era of Sciences. So I am again here to teach you one of the most relevant and relevant topic, that is bioinformatics multiple sequence alignment. So before that we want to request you that please subscribe my channel, that is Era of Science, for latest lecture related to biological sciences and computer sciences, and kindly subscribe it. Okay, so let's start. So first of all I want to tell you something about the sequence alignment. So what is the meaning of alignment? Sequence alignment. Alignment means, as we know, that if we have a car, so we require the front two wheels and back two wheels should be aligned properly so that the car is moving forward easily. Suppose if that wheel is this, the front one of the front wheel is this type and second one is this type. So is it okay for this car, or is it okay for the wheel, for the tire? No. So similarly we are going to align the sequences. Suppose if you have two friends, one name is Akshat, second name is Ashish, third name is Alok, and I have to take my name, that is Abhishek. Okay, so on the behalf of the sequence, I have to select that which one is closely associated with me with sequence, through sequence. So here we see that Akshat, in Akshat A, Ashish A. So A is common. So no. So only one is common. So out of which 1, 2, 3, 4, 5, 6, 7, 8. So 1 out of 8 into 100. So if we cut it, 4 to the 8, 4 to the 8, 4 to the 8, 4 by 20. So 2 and 2, 2 and 2, 2 to the 4, 2 by 10. So 12.5% is similarity between all these four name. If we take, suppose second example I have to take, one name is my Abhishek, second name is Abhinav, third one name is Abhilash, fourth one is Raj. So here common name is A, B, H, I is common. So S no, H no, E no. So A, B, H, I. Out of 8, 4 means 4 to the 8 to 50. Is correct to 50 is the alignment score for these four sequences. Okay, got it? So in this figure we shows that the DNA sequence says that what are you gonna sequence me? Here is the one of the DNA sequence, one of the computer, and this is the analyzer. Because the DNA tell to the system that what are you gonna sequence me? Then computer says that calm down dude, cluster Henry will only analyze you. Yeah, just analyze. Means he feel fear that what the computer and analyzer do with them. So the sequence. So the computer says that only I have to do only sequence similarity, sequence alignment. Nothing more than that. I am not going to cut you. Your sequence is fully for you. I am not be able to cut you, but I have to sequence with the database. So used to measure their, why we are using that sequence similarity. So used to measure their similarity, determine the residue correspondence, Residue means nucleic acid, or nucleic acid means nitrogen bases and amino acid. Okay. Observe pattern of conservation and variability, infer evolutionary relationship. Okay. So types of alignment. There are basically two types of alignment are there. One is global alignment, second one is local alignment. So first of all, I want to tell you something about the difference between global and local alignment. Suppose one sequence is AAT, AT, AT, AG, CD, GG, CG. How much bases are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. This is for example. So if we align the total nitrogen bases, that is known as global alignment. If we cut it differently, suppose 1, 2, 3, 4, 5, 6, 6, 6, 6. So align separately. 6 first we align 6 nitrogen base, then 6 nitrogen base, then again 6 nitrogen base. This alignment is known as local. Local means part wise, and global means whole sequence. Okay, is it clear? Global alignment. A global alignment is a comparison of two sequences over their full length. It assumes that the two sequences are similar and it's attempt to match them from one end to another end. Means one end to another end. Starting is from here and ending is here. So this is global. Okay. And local alignment compare two sequences, but it does not force entire sequence to align. It searches for places that are similar and only align though. Means in a whole sequence, only a part whose sequence are getting get matched, that part of the sequence get aligned with the database, that is known as local alignment. Okay, is it clear? So I have to take one example, that is eye of the tiger. Okay, in 1994, Walter Garen Elterman turned the gene eyeless on in various places of the Drosophila melanogaster. I will show later on how this eyeless gene is on in various places of the Drosophila melanogaster. So this results the eye are going to be formed on multiple places on Drosophila melanogaster. Okay, due to this only. ILS is a master regulatory gene that controls plus minus 2000 other genes for the formation of the eye. ILS on induces formation of an eye. ILS gene, if ILS gene is going to be on, that induces formation of an eye. So it is a very good picture. It shows that this is the eye and it expresses anywhere. This eye can express in the nose, in this cheek, and the head also. Okay. So this is eyeless Drosophila, which does not having any of the eye. And mutant Drosophila melanogaster genes, eyeless turn on. When this eyeless genes are going to be turned on, different eyes are going to be formed. This is the eye of the Drosophila. Okay, did you got it? This is the eyes, or we can say that multiply. So historical homology. Historical homology means there are the different eye containing organism which having eyes, and through this phylogenetic tree we can easily identify that which are going to be closely associated with each other with reference to the eye. That is known as generative homology. Understood? Homeobox, one of the good example is a homeobox. A homeobox is a DNA sequence found within a gene that are involved in the regulation of development, that is morphogenesis of animals, fungi and plants. So sequence alignment, Drosophila melanogaster, Hox homeobox. These are the Hox. Hox is the homeobox for the Drosophila as well as this mouse. So this Hox is this Antennapedia, as we know that Antennapedia, and then bithorax complex, that is posterior, and Antennapedia complex is going to be produce anterior part, bithorax going to be produce posterior part of the Drosophila melanogaster during the developmental stage. Okay, so this is the lab DFD Antennapedia AVDB genes. So this is the common. In Hox3 this is not going to be found. In a fly chromosome this is not found here, but it is found here, absent. This is common. This is common. Means AVD genes is common. Antennapedia is not found in Hox4, but it is found in Hox2, Hox1 and Hox3. So it means the alignment, when we doing the alignment with the Hox2, Hox1, Hox3 and Hox4 gene sequence, we can easily detect the absence gene sequence from the whole sequence of their particular gene. So Drosophila melanogaster packs homeoboxes. Is it not necessary? This is just for your reference point of view. I am not going to define it. So on sequence alignment is the most important task in bioinformatics. It is one of the important part of the bioinformatics. Without this alignment analysis, we cannot do anything. We cannot do any analytical part from the bioinformatics. So I would like to prefer you that you please go through this video, this lecture, so that you can at least you can identify how we can align a sequence and what is the benefit for that sequence alignment. So on sequence alignment, for why the sequence alignment is important. So first is prediction of function, okay, database searching, gene finding, sequence divergence, sequence assembly, okay, prediction of function. Suppose I have X gene and I want to identify the function. So we have to do alignment this unknown sequence with database whose sequence is known. Suppose after alignment we found 100% identity. So we can easily say that this X unknown gene can do same function as this Y gene 2, whose function is going to be registered in database. So database searching means, suppose you isolate DNA, amplify with random or specific primer, and after that you send it for sequencing. The sequencing person give you the 326 base pair sequence for your analysis. So how you can identify that this 326 base pair performs this function? So you simply do a blast so that you can search the similar type of sequence from the database, either from gene database or protein database. If you doing protein sequencing, then you have to go for protein database. If you are doing nucleic acid sequencing, then you go for gene sequencing. Gene finding, how you can find by the help of this alignment you can find the gene. Then sequence divergence, how much your sequence is going to be diverted from the original one, means mutation analysis. This means mutation analysis. Then sequence assembly means, suppose there are one sequence or chromosome, how this ABCD are aligned. Suppose this is the sequence of this ABCD gene. So the sequence is going to be found through this AC, through the alignment results. Okay. So on sequence similarity, homology means the gene that divert from a common ancestor gene is called homology. These are the basic terminology which are going to be used in alignment. One is homology, second one orthology, and paralogy. Orthology means homologous gene in different organisms. Paralogous genes are homologous genes in one organism that derive from gene duplication. Gene duplication means one gene is duplicated in multiple copies that therefore free to evolve and assume new function. So homology and paralogy means this is the one of the ancestral tetrapod. So these are going to be means evolved. So bird hand is like this, whale tail is like this, horse hand is like this, bat hand is like this. So these are going to be evolved, and this is the hand of human. So this is the paralog, means perform same function in different species. Understood? Means all this organ having the same function, that is like hand in human. Okay. So similar type of things are going to be shown here. Here also. So sequence alignment causes for sequence dissimilarity. If suppose if anyone has any sequence have one mutation, that is this shows that ATA to AGA means ATA to AGA means in place of T, G is going to be inserted. Okay. So this is the mutation, that is insertion. This is T to G means transversion mutation. Insertion means A to AGA means G is going to be inserted in it. Deletion means suppose we have ATG, ACTG, then AC and G. So this T is replaced. Here G is inserted. Here in place of T, G is located. So this is the sum of the dissimilarity found. Indel means an insertion or a deletion. Sequence alignment, global and local. Find the similarity between two DNA sequences by finding a good alignment between them. The biological problem of the sequence alignment. What is the problems we face for after doing the sequence alignment? Suppose this is the DNA sequence. Okay, DNA sequence 1. This is the DNA sequence 2. So this is the 1 and this is the 2. Okay. So T, C, T, T, T. This is misplaced. This is not matching. This is gap. And rest other are common. So this is going to be aligned. This blue bar shows the alignment, means same sequence are there. So sequence alignment is the arrangement of two or more sequences highlighting their similarity. The sequences are padded with gap, that is dashes. This is the gap, so that wherever possible column containing identical characters from the sequence involved. Understood? So there are some of the algorithm for alignment, that is Needleman-Wunsch algorithm, which is pairwise global alignment only. It does only this. Smith-Waterman pairwise local or global alignment can do this both. BLAST means basic local alignment search tool, that is pairwise heuristic local alignment. Okay, pairwise alignment means method are concerned that way with finding the best matches piecewise local or global alignment of protein or DNA. Typically the purpose of this is to find homologous of a gene or gene product in database of known example. This information is useful for answering a variety of biological questions. Number 1 is the identification of sequence of unknown structure or function. Number 2 is the study of molecular evolution we can do by the help of pairwise alignment. Okay. Global alignment, as we already discussed, that whole sequence we can align through the database through global alignment. So global alignment find the global best fit between the two sequences. Example, suppose I am going to discuss one of the best example here I shown. The sequence S is Viva Las Vegas. Okay, here Viva Davis, VIVA LAS. I already write it. Viva Las Vegas. Okay. So here the common is S and T is the name. V is common. I, V, A. It is mismatched. This is all again matched. This is not. This is matched. This is matched. So 1, 2, 3, 4, 5, 6. 6 are going to be matched in 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay. This is the insertion deletion, gapping. Okay. So if we found the alignment score for these two, so we can find 6 by 12. 12 is the total maximum into 100. Means, okay, 3 1 is 3, 3 2 is 6, 250. So 50% matching are there between these two, Las Vegas and Dallas. Okay. Second one is Needleman-Wunsch algorithm, which perform the global alignment of alignment on two sequence, that is S and T in the applied aligned protein or nucleotide sequence. It is an algorithm to example of dynamic programming, and it is guaranteed to find the alignment with the maximum score. Means Needleman-Wunsch algorithm shows the maximum score in the alignment process. Of course, this works for both DNA as well as for protein. Alignment score function is the cost of aligning two symbol x1, y1 is scoring function, that is x1, comma y1. So the cost of entire alignment is that m is equal to summation of CIL is in the sigma x1 and y1. This is the alignment cost. A simple alignment function, I am discussing it later on, so no need to put your emphasis on that. I am going to discuss it separately so that you can understand what I want to say, what the specific matrix or what a specific alignment going to use, or specific programming for aligning the two sequence, two or more sequences. So scoring function, how we can find the score? So this is MA, that is 7 matches, 1, 2, as I already discussed there, 1, 2, 3, 4, 5, 6, 7. Sorry, there I write, at that place I write 6. No. 1, 2, 3, 4, 5, 6, 7. Sorry, this is wrong. So 7. I am going to discuss it now. This, the total 7 matches plus 2 mismatch plus 3 gap. This is 1, 2, 3. Mismatch is 1, 2. Okay. So 7 plus 2, 8, 9, 10, 11, 12. Total is 12. So 7 minus 2 minus 3 is equal to 2. So this is the score, cost of the alignment for the two sequences. Okay. So optimal global alignment between two sequences S and T is the alignment that maximize the total alignment score, MA, overall possible alignment. Okay. So here A is equal to argmax MA. Find the optimal alignment. A, looking the combinatorial optimization problem. Generate all possible alignment, compute the score M, select the alignment A with the maximum score M. So next one is local alignment. Local alignment method find related region within sequence. They can consist of a subset of the character within the each sequence. For example, position 20 or 40 of a sequence A might be aligned with position 50 to 70 of sequence B. Suppose one of the two of the sequences are containing 100 base pair. So one sequence having 20 to 40, this much sequence are going to be aligned with sequence B 50 to 70. This is going to be matched. So this 20 to 40 and 50 to 70 is the aligned locally. So that's why this is known as local alignment, okay, which is not possible through global alignment. So next algorithm is Smith-Waterman. Smith-Waterman is the scientist who discovered this algorithm in 1981. Is for determining similar region between two nucleotide or protein sequence. Smith-Waterman also dynamic programming algorithm to and improve on Needleman-Wunsch algorithm. It has desirability property that is guaranteed to find the optimal local alignment with respect to the scoring system being used. It is a specific, this Smith-Waterman is the modified version, or we can say that the some of the algorithm programming taken through Needleman-Wunsch, and this is the improved alignment algorithm from Needleman-Wunsch to Smith-Waterman in 1981. Okay, it is used for BLAST algorithm only. Okay, sequence alignment meaning is to use to study of the evolution of the sequence from the common ancestors such as protein sequence and DNA sequence. Mismatch is the alignment correspond to mutation and gaps correspond to the insertion deletion. Means mismatch means, as here we show, sorry for again going to there, mismatch means this LD S, sorry no SS, IA and VE. This is mismatch through mutation. Again this LD mismatch repair mismatch, this is mismatch. These two are going to be through mutation in the, and this gap is indent, or it means insertion or deletion. Okay. Sequence alignment also refer to the process of constructing significant alignment in the database of potentially unrelated sequences. So, statistical analysis of the alignment. This histogram shows the randomly generated string using permutation of the original sequence T, as I shown, that is Las Vegas. Okay, original sequence S is here, and this is the mismatch or matches of the sequences. So that's all for today lecture. I just want to tell you that please share, like my channel, that is Era of Sciences. Kindly click on bell so that when I will go to upload the lecture, you can easily retrieve from there. So thank you very much for today's class. Good night, friend. Again, I am going to upload this BLAST and multiple sequence alignment in my next lecture video. Thank you.